Okay, here's our second podcast for Standard 1.3. We're going to take a look at how enzyme activity is regulated in living things. So when we start to think about regulating our enzymes, we have to remember that enzymes are only going to work at certain conditions. And that is why living things need to maintain homeostasis. So all enzymes have optimum temperature and pH at which they will work, and if they are not at that optimum temperature or pH, they will not work correctly. And that's why humans have to be around 98.6 degrees in order to function correctly, and we also have to be around a neutral pH because our enzymes have to work right, and if our enzymes aren't working right, then none of our cells can work correctly. So proteins will be denatured if conditions are not perfect. And if they become denatured, they will stop working. What denatured means is that they will fall apart. So if you look at this picture here, if you have a protein, remember enzymes are proteins, and you look over here, native forms of the protein, we talked about how proteins have this quaternary or tertiary structure where they fold up on themselves. In a denatured situation, they will unwrap, unfoil, and they can no longer function correctly when they are not in their native form. So it's a lot like when you cook an egg. You know how the egg white turns white when you're cooking it? That's actually because you're denaturing the proteins. Sometimes after denaturing, proteins can go back to their regular form, but very often they cannot, and then you have to make new ones. So if proteins are denatured and they're not working correctly, then the enzyme is not going to work the way that it needs to work. So we're going to look at how enzymes are affected by changes in temperature. So here we have our enzyme. And remember, the active site of the enzyme is only going to fit specific substrates. So here, this substrate will fit here. Okay, and if I try to put this one in somewhere, it's not going to work. It gives me that lovely little sound. But here, this one will fit in. And so when they're there, we've lowered the activation energy, and now we've made our new product. So we can take a look at what will happen to an enzyme if it's not at optimum temperature. So here we are at 17 degrees Celsius, which is a little below where we would want to be if we were in the human body. So it's an 84% effectiveness if you are looking at an enzyme at this level. If we reset it and we go up to where normal human temperature would be, we're doing a little bit better. We're at 98%. So the normal human temperature is about 21 degrees Celsius, and there we're working at 98%. Now watch the enzyme. If I go really hot, look what happens to the enzyme. It gets all stretched out. So here we are super, super hot. The enzyme is now denatured. So if I try to fit those substrates into that enzyme, they can't fit at all. It doesn't work even a little bit. Okay. If I go a little bit too hot, let's say like this would be a pretty decent fever going on, right? They can get in there, but they don't really like it. Okay, they don't fit very well, and that's only a 25% efficiency. Same thing happens if we go in the other direction. If we get really, really, really cold, the enzyme is going to kind of scrunch up, and the substrates are going to have a really hard time fitting within there. And remember, chemicals also move slower when it's colder. Okay, so they might get in there eventually, but it takes a really long time, and it's only 14% effective. Okay, and even if we go a little bit warmer, still we're not going to be perfect. So this enzyme is going to work best at a particular range. So if we go back to our PowerPoint here, so we just talked about how we had denatured our enzyme there. It changed shape. And what ends up happening is for a typical enzyme, like here is an enzyme that is working the best at around 40 degrees Celsius. That's about the peak. So somewhere between 30 and 50 is the best temperature range for this particular enzyme. If you go too high over, okay, the rate at which the reaction works falls very fast because you have completely destroyed the enzyme. If you go too cold, it's a slower change in the rate because you're really just making everything slow down and the pocket's getting smaller, but the enzyme and the substrates can still fit together a little bit. But it works at the absolute best right around 40 degrees Celsius. That's where this particular enzyme, whatever it is, would work at the best. Okay. Now we also have other ways of regulating our enzymes. Sometimes there are inhibitors that we will have within our body, and these are going to help to control our enzyme reactions. So sometimes there are inhibitors that are competitors. So they look like the substrates, but they don't actually cause a reaction to happen. So what they'll do is they keep the reaction under control because they will fit into the active site so that the substrate cannot. A lot of times medications will do this. A lot of different kinds of medications will actually act as competitors to our substrates. And that can be really important in some cases when you have enzymes that are actually overacting. So those kinds of things can happen with these inhibitors. You also sometimes will have enzymes that have to be activated before they can be used or before anything can happen. So in this case here, we have our substrate and our enzyme. And if we're going up in this direction, so here we have our enzyme and it's not in the right shape right now. And we need it to get relaxed before it can accept the substrate. So an activator will come in and bind to this pocket, and that will make it get into the correct shape, and then the substrate can bind to it. So activators also can help kind of regulate the way that enzyme reactions occur, and we also do have medications that act as activators for enzymes as well. All right. One last thing, when we talk about enzymes, there are some tricks to naming them. Okay? Enzymes are very often named for the reaction that they catalyze. So whatever the name of the reaction is that they are going to be speeding up, that will be part of their name. So lactase is an enzyme that helps to break down lactose. Lactose is a sugar, lactase is going to break it down. It sounds the same, right? You can see it. Almost all enzymes also are going to end in the suffix ase. So anytime you see ase, think enzyme. And remember, when you see ose, that is a sugar. Okay, so examples of this: amylase, DNA polymerase, catalase. These are all enzymes, and I know immediately that they're enzymes because they end in ase. Then not only do I know that that they're enzymes, I also can then say I know they're proteins because all enzymes are proteins. So they're all connected. All right, so let's summarize everything that we learned in Standard 1.3. So remember, many chemical reactions require energy to occur, and that energy that is needed to get a reaction started is called the activation energy. Okay, enzymes are proteins. Don't forget that important piece of information, that are going to lower the activation of a reaction, and that's going to make the reaction happen faster. So when you make it so that it needs less energy to occur, the reaction will then go faster. And remember also that enzymes are highly specific, so they're only going to work with certain substrates. That's because of the active site. And they also have to work at optimum conditions. They have to have certain temperature and pH in order to work correctly. And if your body's not in homeostasis, your enzymes will not work correctly.